Welcome to the second session of day 2 of the PGC Pathways to Therapeutics meeting. We now have a series of five plenary talks, keynote talks. It's a pleasure to introduce the first speaker, who's Prof. Joel Gelernter. He's a professor of psychiatry, genetics, and neuroscience at Yale School of Medicine. Joel has done some famous work in substance use disorder genetics. He has been involved in large-scale and led large-scale collaborative consortia working in Asia, Taiwan, Thailand, and China. But today he's going to talk to us about the Million Veterans Program, which is one of the most exciting programs in psychiatric genetics at the moment. Okay, with that, I'll stop sharing, and Joel, if you could.
Okay. Yeah, sorry, I'm still trying to find the sharing link.
Normally, if you hover with the mouse at the bottom of the screen, then it should occur again, like share screen at the bottom. Yeah,
got it. Thank you. So thank you, Jerome and KCL crew and organizers for the opportunity to talk to you all about the Million Veteran Program. First, my disclosures, which are unfortunately very brief. So the Million Veteran Program is described as on the screen on the official MVP website. The link is given there. It's a national US voluntary research program funded entirely by the US Department of Veterans Affairs Research and Development. The goal is to partner with veterans who receive their care to study how genes affect health, with a particular focus on behaviors and traits that are prevalent among veterans. So to do this, the MVP is building a large medical database. Originally, the plan was to collect data from 1 million veterans, but the idea now is that they're going to reach 1 million and then keep going. So, so far they have recruited 825,000 subjects, and that's really great, but it's going to get better. Veterans who are participants are assured of the security of the data and of the relevance of the work that's to be done to veteran-related health issues. There has been a paper published describing the background of the MVP by Mike Gaziano, and the reference is on your screen now if you want to read more about the background. The MVP has several characteristics as a gene mapping resource that sets it apart from and makes it different from most of the other databases that are available and that we're used to using. The sample size is large and growing. The assessment is homogeneous. There is considerable representation from non-European samples. So there are advantageous things, but it's also somewhat disadvantageous that the sample is mostly male. There are data available from the electronic health record, including many longitudinal assessments from the EHR. For example, sometime over a decade ago, the VA mandated regular assessments with the AUDIT-C, which is an alcohol use quantity frequency measure. So many patients have not just one AUDIT-C, but numerous measures. And the MVP also collects data from self-report surveys. Compared to other samples, participants in MVP tend to be comparatively old and sick. They are identified for possible recruitment because they've used VA health services. 55% of them are between the ages of 50 and 69. In this talk, I'm going to focus on some of the utility of the MVP for gene mapping for psychiatric traits, although it's a marvelous resource for other traits too, lipids, cardiac function, others. We've done work in substance use disorders and PTSD and anxiety traits. And I'll point out that there are several other groups that are also accessing the MVP for studies related to other psychiatric traits. Phil Harvey is working in schizophrenia and bipolar, Nate Kimbrell in suicide, Panos Roussos, Hank Kranzler and Amy Justice. Today I'm going to pick a few examples. I'm going to discuss alcohol use disorder and audit C, opioid use disorder, and anxiety traits. So the first individual study I'm going to discuss is one that came out last fall on the subject of alcohol use disorders and audit C. And this study involved 274,000 participants, and we were able to include data from 5 different population groups with substantial contributions from European and African ancestry subjects. The 2 main traits are alcohol consumption measure. So audit C comprised of 3 questions. How often do you have a drink containing alcohol? How many standard drinks containing alcohol do you have on a typical day? How often do you have 6 drinks or more? So that's one of the 2 traits. The other trait is alcohol use disorder by ICD-9 or ICD-10. Prior to this, we also published a GWAS on a survey question, maximum habitual alcohol use. I'm not going to discuss that study today. The sample size was smaller and it was not as well powered, although it also had some interesting results. So this paper, Kranzler et al., came out in the fall, and here's the reference. So at the time we did the study, there were a little over 500,000 MVP subjects enrolled. Of these, 350,000 had available genotype data, and that's still the case. There hasn't been a new data release for quite a while, although we're expecting one end of summer, early fall. And just to look at who's in the sample, we were able to include over 200,000 European ancestry and 56,000 African ancestry. There are much smaller numbers available of Latinos and East Asians and South Asians. We included those in the publication, and we are making the summary statistics available too. And the reason we included those samples, although they're rather small, was primarily to have a means to enumerate the summary statistic data from the MVP, which can sometimes be quite hard to do, to make this data available for other meta-analyses. Here is the result for the audit C meta-analysis. And you'll see that consistent with expectations, by far the largest number of results are available from European ancestry subjects, but there are some significant results also from African ancestry and Latino. And even in the rather small East Asian sample, there was a significant result for a SNP in high LD with ALDH2, which is a very well-known alcohol risk locus in some Asian populations. So this analysis and many of the other analyses that I'm going to show were done by Hong Zhao, data analyst working with PGC and MVP and many other studies. So that was the result for audit C. Here are the results for alcohol use disorder. And it is perhaps evident to the eye immediately that there are some differences, and there are somewhat fewer significant hits for alcohol use disorder. It's somewhat interesting to look at the overlapping and independent genome-wide significant signals for these 2 phenotypes. So for alcohol use disorder, 10 loci. For audit C, 13 loci. This is in the European ancestry subset of the sample. And only 5 of these overlap. So the alcohol dependence trait, which is the one that we regard as pathological, is showing some genetic differences with audit C, the alcohol quantity frequency measure, which picks up a lot of variation in the normal range. And indeed, when we look more closely at the genetic correlations by LDSC for audit C, quantity frequency measure, and AUD, alcohol use disorder, the pathological state, what we usually view as the disease state, we see that there are some striking differences in pattern for LDSC. And by the way, quite similar results were obtained by Sandra Sanchez-Roig and Abe Palmer and their collaborators in UK Biobank, looking at the different constituents of the audit C as here, and the audit P, which is the problematic alcohol use assessment part of the audit. So just to point out some of the differences for anthropometric traits, for traits like BMI and obesity, we see negative correlations with audit C, but not with alcohol use disorder. For coronary artery disease, alcohol consumption in the normal range has negative correlation with coronary artery disease. But when we look at psychiatric traits, the correlations that are there, for example, with MDD and audit C, tend to be negative, but alcohol use disorder, alcohol dependence, is positively correlated with a range of psychiatric disorders, including depression, bipolar disorder, and schizophrenia, and of course, alcohol dependence in other samples. So our conclusions based on the MVP studies of alcohol use disorder and related traits is that we have multiple loci, some that contributed to all traits. I didn't show the max ALK data, but some of the variants that were common to audit C and AUD were also common to maximum habitual alcohol use, some associated with only one trait, at least at our current level of power. Presumably as samples get larger and larger, we'll see these not as single trait associations, but as loci that are differentially associated. But nevertheless, it's still fairly striking at this stage that variants in CNS genes seem to be more important for alcohol use disorder, and in the context of very heavy drinking than in the context of normal range drinking. There were more findings for AUD, quantity frequency, and audit C than for our earlier maximum alcohol study, reflecting larger sample size that's available with EHR versus self-report data. Some of the strongest loci were alcohol metabolizing enzyme genes, and these have been discussed previously in the meeting by Arpana Agarwal and Alex. These are unusual in psychiatric genetics in that these strong associations have survived from the candidate gene era. So these are really strong and at this point really reliable, and they appear to be common to a strong range of alcohol-related traits. And just a final message from this study is that huge samples get you a lot more, but a lot of times we've been willing to accept less distinct phenotypes that we think are related to the traits that are really of interest. So when we started the work, we thought quantity frequency measures like audit C would be good proxies for pathological states like AUD, alcohol use disorder. But that hasn't been the case at all. And we've seen similar patterns for other substance use traits. And we might expect to see them for other psychiatric traits as well. Following on the AUD audit C trait, we then did a meta-analysis for problematic alcohol use using all the data available from the MVP, from the PGC, Walters et al. Alcohol dependence meta-analysis, and UK Biobank. Whereas for the MVP work, we were able to include non-European populations. For the meta-analytic work, we are now restricted to focusing only on European samples because of the relative lack of other populations to study. So this work is available for your immediate perusal on bioRxiv, and it's just recently in Press in Nature Neuroscience. And this analytic work also was done almost exclusively by Hong Zhao. This is a quick look at the design of the study, and I'm going to focus on just a few elements of this. First of all, compared to the first MVP AUD and Audit C analysis, we are now adding in phase 2 samples from MVP. From the latest data release, 65,000 new subjects. So the meta-analysis within MVP provided the core of the analysis. Then we meta-analyzed in the alcohol dependence data from the PGC publication, Walters et al., which came in very late 2018. And finally, we meted in Audit P problematic alcohol use from the Sanchez-Roige paper with Abe Palmer. This was less strongly correlated with AUD than alcohol dependence, but there's still good evidence that that's primarily what it indexes as opposed to quantity frequency. We also included in the paper an MTAG analysis with GSCAN data with drinks per week, but I'm not going to discuss that today, and I'm also going to discuss only a limited number of post-GWAS analyses. So what you see before you now is our cover concept for Nature Neuroscience. I don't know if they'll take it or not, but what this shows is an alcoholic beverage, which I like to think of as a Lefroy single malt, indicating with shiny glimmers alcohol risk loci on a karyotype. So in the main meta-analysis, we're now able to include 435,000 subjects. And you can see, as has been the case for most alcohol studies, ADH1B is by far the strongest hit. But now we have, instead of 10 loci, 10 independent loci in the original MVP GWAS, we have now 29 conditionally independent loci. And the findings are quite a lot stronger, and this means that we can take them quite a lot further in our next step post-GWAS analyses. Here is the PheWAS data from BioView done with our collaborators at Vanderbilt. And very much as expected, the strongest associations are with alcohol use disorder, tobacco-related disorders. Consistent with the LDSC results, we see also PheWAS significant associations with major depression, bipolar, anxiety disorders. But there's some other interesting stuff too. Here pops out to our interest, but not to our surprise, alcoholic liver damage. And then we also have some pulmonary traits popping up as significant: chronic airway obstruction, bronchitis, and emphysema. And here is the LDSC result from problematic alcohol use. And the findings are very similar to what we saw previously with AUD, but again, they're quite a lot stronger. The correlations tend to be a little greater, and the variance is tighter. There aren't any huge surprises here. Strongest findings associated with drinking and smoking, and then cannabis use. And again, things associated with risk-taking, like number of sexual partners and general risk tolerance. Negative correlations with subjective well-being and age of initiation of smoking. So from the PAU study, Zhao et al., it is again a meta-analysis for problematic alcohol use, 29 conditionally independent risk loci, 19 of which are novel. We reported, but I'm not discussing today, functional enrichment in brain regions and MR analyses from Hong, and then genetic correlations with psychiatric, medical, and health outcomes. Just to bring to the forefront again, the fact that we needed to focus on European ancestry samples for this study, because that was really all that were available to us. And collection of non-European subjects is really critical. We couldn't increase the number of African ancestry subjects appreciably compared to our initial GWAS. Next trait I'm going to focus on with MVP data is opioid use disorder, or OUD. Until COVID, the opioid crisis was getting a lot of press in the US. Opioid overdose and opioid use being at epidemic levels in the US. Previous GWAS for OUD and related traits have identified risk variants, but there was none with a clear replication. They all had limited sample sizes because there's a limited number of human subjects available for OUD. Compared to some other psychiatric traits, the prevalence is relatively low. And even from large biobanks, the harvest of OUD subjects is comparatively small. Another issue with studies of OUD is that phenotypic definitions have varied. So you have OUD, or chronic opioid use, or injection opioid use, to define cases. But I would argue that it's an equally important issue how controls are identified. So for a trait like opioid use, where many people who are exposed to opioids ultimately become dependent, I think it's important to consider that unascertained control groups that include subjects who have not been exposed to opioids are more legitimately considered diagnosis unknown than control groups. To know someone is really a control with respect to opioid use disorder, you also have to know that they are exposed to opioids and have had the chance to express whatever genetic liability to OUD they might have. So we have used these models for our studies, and we used it for the MVP study. A recent paper in Molecular Psychiatry, the PGC study of OUD, has reinforced this even further by looking at opioid unexposed versus opioid exposed subjects. We wish that study was better powered, but I still commend it to your attention, particularly with respect to the issue of how control groups are defined and constituted. So to look at OUD in European Americans, and again, there weren't enough African Americans to study in the MVP sample, we defined cases by electronic health record, and we were able to get about 8,000 cases, which is a decent number, but not huge. But we were also able to do something very interesting with our control definition. And that is, there had been a previous analysis by Chris Wrench looking at prescription opioid use trajectories that defined different groups of subjects based on what happened to them after a 7-day exposure to prescription opioids. And just in short, some tended to increase their dose, some had varying degrees of dose increase, but there was also a subset that was exposed to opioids and then didn't tend to increase their use over time. So we use this group of subjects, which are called Trajectory 1, people who are exposed to opioids by prescription for 7 consecutive days, had more than 6 months of follow-up, and who didn't tend to have increased dose to define our group of exposed controls. So by these definitions, we had 8,500 cases and 71,000 opioid exposed controls. There were not enough cases in MVP to attain statistically significant results from that sample alone. We had to meta-analyze with our local sample, which we call Yale Penn, sample we started collecting in 2000, and with SAGE. This is a sample that we obtained via dbGaP, where Beirut is the main PI of that subject study. So we ended up with over 10,000 cases for this analysis. And putting everyone together, we now have one genome-wide significant hit. But it's quite an interesting one because it maps to a quite well-known functional variant at the mu opioid receptor locus. So this is a pretty well-known, pretty well-studied candidate gene. The candidate gene studies previously tended to be underpowered and gave very inconsistent results. But there were quite a lot of previous studies going back to the 1990s supporting a true functional effect for this variant. Going back to the study from MJ Creek and Lei Yu and PNAS, and here's a study from Wolfgang Sadi's group from the early 2000s. There have been a lot of studies in different model systems. We were also able to replicate this finding looking only at the one lead variant to preserve power. We replicated in newly genotyped Yale Penn subjects and also in UK Biobank using the proxy phenotype of buprenorphine treatment. Buprenorphine is generally used to treat opioid dependence. It can also be used to, as pain treatment. But by the assumption that most of these subjects had opioid use disorder, we obtained a barely statistically significant replication in UK Biobank and similarly in Yale Penn, leading us to a meta in the 10 to the minus 10th range. For the first time for OUD, we're now able to look at the genetic correlations by LD score regression. We find correlations with substance use traits and also, fortunately, with opioid use. So powered mostly by the MVP, we're now able to report genome-wide significant association with OPRM1, replicated genetic overlap with many traits, possible causal effect on OUD, that's work I didn't discuss. And again, we still have rather modest sample sizes for OUD. Recruitment of more subjects is absolutely critical. Some are going to come from other biobanks, but in particular, we argue that targeted recruitment of non-European ancestry subjects is a really critical next step. We've looked at PTSD in MVP. This is one of our most exciting initiatives, we believe. Murray Steen gave a great talk on that yesterday. I'm not going to discuss that today. I'm going to discuss very briefly some of our work with anxiety. Common trait, it hasn't been a major focus of funding agencies because they have limited funds and they have schizophrenia and bipolar disorder to focus on. So biobanks are particularly useful for traits like this. This is work done primarily by Dan Levy, postdoctoral fellow in my research group. We looked at a continuous trait, GAD2. These data are also available, by the way, in UK Biobank, and also a binary phenotype. These are circus plots showing results. We were able to look at AAs. We did have one genome-wide significant locus, but as is usually the case, due to greater power, we saw more in European ancestry. In the interest of time, I'm going to skip over most of these results. They're published in our American Journal of Psychiatry paper in January. I'll just highlight two of them. One is SATB1, which modulates expression of CRHR1, is related to CRHR1, corticotropin-releasing hormone 1. This has popped up in our studies of alcohol use and PTSD also, and is clearly related to stress responsivity. And another locus of great interest that was significant in the study is estrogen receptor A. So this has frequently been linked with anxiety traits in humans and anxiety-like behaviors in animal studies. We also wonder if it's related to the strong female predominance for anxiety disorders that's observed epidemiologically. And bear in mind that the MVP is 93% male. So this is not explained by the female predominance in our sample, just a relationship with female sex hormone. And I will skip over the MT COJO and go to summary about the MVP and what it's good for and some of the characteristics. First, it's large and getting larger. The current goal is maybe 2 million. The subjects are relatively ill and relatively low socioeconomic status, and that enriches it for information content compared to some higher SES samples like 23andMe and UK Biobank. There's useful data from the EHR, but not only that, the useful data goes way back to the 1990s. And additionally, there's good self-report data from about half of the sample. It's very diverse compared to other large samples. We have a problem with our array coverage in MVP, which is much better in Europeans and non-Europeans. That is being addressed by application of a supplementary array enriched for African ancestry content in all non-Europeans in the MVP sample. But at the moment, it's hard to gain access to the MVP, and it's hard for MVP investigators to share summary statistics pre-publication. There's a way to do it, but there is a somewhat cumbersome mechanism. However, I just learned at a meeting yesterday that access should improve soon, probably within the next year. And there's also a movement to make our summary statistics data available on publication still via dbGaP, but now in the non-protected area. Presently, you now have to go through the usual dbGaP application process. We would like to have them available for immediate download. So finally, MVP, less well known than other large biobanks and consortia, but it has unique advantages, has already proven useful in mapping psychiatric and SUD traits. Finally, I want to acknowledge my very large cast of collaborators, particularly Murray Steen, Renato Polomonti, and the people in his lab, Dan Levy, and through the MVP Beta Project, Hong Zhou, and also Hank Kranzler, Amy Justice, Rachel Kember, and MVP supported by the US Department of Veterans Affairs. And with that, thank you for your attention, and I'll turn it back to the moderator.
Okay, thank you very much, Joel. That was a great talk and exactly on time. So, well, thank you. We have time for one question, I think. Are there any questions from the audience?
Currently, no question from the audience.
Okay, so Joel, MVP is truly a remarkable project. I mean, you do have a big preponderance of male cases. What do you think the fundamental differences are between MVP and, let's say, other hospital-based system recruitment in terms of psychiatric disorders? And can it be used to address some of the lack of male recruitment that we see in other studies?
So are you asking if the predominance of males is an advantage compared to other studies with the predominance of females? I certainly think when we want to meta-analyze them and look at sex-specific risk loci, which we all expect to be there and which we've had a hard time identifying, yeah, it could be an advantage in terms of balancing out what's available worldwide. We wish we could address females more in the MVP. The other side of that, the advantageous side, is that it makes our sample in the MVP somewhat more homogeneous, increases our power for male-specific risk loci.
Thank you. Okay, I'll hand over to Topher to introduce the next speaker.
It's my pleasure to introduce Professor Matthew Hotop, who is the Vice Dean of